All right, welcome everybody to a new Xylella Files seminar. Thank you for joining in. And it's my pleasure today to introduce the speaker, Miguel Román Écija. So Miguel is a Ph.D. student in the Institute for Sustainable Agriculture of CSIC in Córdoba, Spain, under the direction of Blanca Landa and Juan Navas. And he's pursuing a Ph.D. in agronomy, food, forestry, and rural development engineering in that institute, and he is getting ready to defend his Ph.D. soon. And I know Miguel for many years. So Miguel, when he started working on this in 2017 and Ph.D. in 2018, and he joined our lab for a few months back then, and I'm very happy to see how he has evolved, and he's been doing very interesting research in the biology, ecology, and epidemiology of Xylella fastidiosa in Spain. And he's going to tell us about his research over the last year. So thank you, Miguel, for your talk, and we're looking forward to learn more about your research.
Thank you very much, Leo, for the presentation. And thank you for giving me the opportunity to present my research work to accomplish my PhD thesis here in the Institute for Sustainable Agriculture. And well, as all you know already, I'm going to only to do a brief introduction about Xylella fastidiosa. That is going to be related with some of the objectives of my PhD thesis. First of all, these bacteria have a high genetic diversity. Also, it is divided in 3 main subspecies and 19 sequence types. Also, is able to infect more than 700 of host plants and cause disease in very important crops. Also, although its origin from the American continent, it has been adapted to different climates. And since the first detection of this pathogen in Europe, in Italy, in 2013, farmers, researchers, and stakeholders raised the concern about the bacteria in the European territory. So my PhD main objective, let's say, is to generate the general knowledge about the new genotypes isolated from Europe. And I usually like to explain my objective, the objective of my PhD thesis using the triangle of the disease, because the thesis touch the three main areas. Well, I'm going to in each objective, I'm going to explain a little of the research we have performed. And for the first objective related with the pathogen, we have characterized phenotypic traits related with colonization and virulence, and also perform a genomic comparison. In this work was mainly performed during my stay in our university under the supervision of Leonardo. Thank you for his team and him. I knew all that I have to learn about Xylella at the beginning. And also the work of the research I'm going to explain this research was already published, and as I perform another seminar explaining this research in more detail, I'm going to explain only the main information. So in this work, we have compared two Xylella fastidiosa strain belonging to subspecies multiplex and isolated from the demarcated area in Alicante in Spain, isolated from almond. Both strains share more than 99% of the chromosomal genome, and the main difference is the strain ESBL harbor 2 plasmid, one big plasmid, another small. So the objective of this work was the phenotypic and genotypic characterization of traits associated to infection and disease development of the two Spanish strains to find out if these differences are due to the plasmid content. So also we use reference strain from United States, from fastidiosa and multiplex subspecies, and we have performed a battery of experiments to characterize the phenotype. We have studied the growth, planktonic, biofilm formation, and aggregation. Also, we have used microfluidic chamber to study the cellular behavior, motility, and adhesion. Also performed twitching motility to study pili morphology, and also tobacco pathogenicity in greenhouse. And the summary of the results are here. And between Spanish strains and the reference strain from the United States, we can say that the reference strain have more ability for biofilm formation in general, and more twitching motility, and also had a higher virulence in tobacco. But despite the similarity of the two Spanish strains, it has also some differences. For example, in the twitching motility and the adhesion force to the microfluidic chamber. So we want to characterize these two strains by its genomic, and we perform several analyses, and also compare with the reference strain Temecula for some of the analysis. What we observed was some unique genes, mainly due to the plasmid content in the ESBL strain, but also unique genes in the chromosome of both strains. And when performed the deleterious analysis, well, not the deleterious, the analysis of the SNPs, we observed that we obtain deleterious function in the motility compared with Temecula, and also deleterious position compared with adhesion. Well, in a second objective, we want to characterize the pathogenicity and the colonization pattern in several crops. In this first experiment, also in collaboration with Leonardo de la Fuente and Dr. Deepak Chandra, we have inoculated seven European strains and the reference strain from the United States that is very virulent in blueberry. And also I'm going to go very quick in this one because it has been presented in other seminars. So the main result we have obtained is that the strain EVIA 5901 has the same virulence that the reference strain of the United States, and the rest of European strain did not cause severe symptoms. And also, interestingly, those virulence was related with a higher planktonic growth. Well, in other experiments, well, in this experiment, we have inoculated 3 olive cultivars widely cultivated in Spain, Picual, Hojiblanca, and Arbequina, and it has been inoculated with 5 Xylella fastidiosa strains isolated in Europe, 4 from Spain, and the reference strain from Italy, the Don. Well, and the objective of this research were 2. On one hand, to assess the infectivity and pathogenicity of these Spanish strains on these olive cultivars, and taking advantage of the inoculation of these plants to develop and validate a handled proximal sensor-based detection system for early detection of Xylella in olive trees. And this work, I'm happy to say that was affected in plant disease with minor revision during the last week. To address the first objective, we have performed several sampling. In this case, at leaf level, we have taken leaves at the inoculation point at 5, 10, 15, and 20 centimeters from the inoculation point, and at 1, 6, 12, and 18 months post-inoculation. And also, we have performed a destructive stem sampling at 9 and 42 months post-inoculation at the end of the experiment. In this case, we have taken several sections of the stems below the inoculation point and at the inoculation point, but also at 10, 20, 40, and 80 centimeters. from the inoculation point, depending of the time of sampling. And to detect Xylella fastidiosa in leaves and stem, we have performed the duplex, the protocol of the duplex QPC. Well, and the main results we have obtained was we had obtained a successful inoculation at the inoculation point and in leaves. In all the treatments performed, and also in this plot we can observe a higher frequency of detection in the strain isolated from Écija belonging to subspecies pauca. Also, if we observe the frequency detection over time, we observe in general a decrease of the frequent detection. And the analysis also showed that the main effect was due to the Xylella fastidiosa strain, and not the cultivar or the time. Also, we have observed a higher number of positive in the donor and the strain of subspecies pauca from Spain. Well, in the sampling at stem level, we observe in general a higher number of positive compared to leaves, and also across the several stem sections. Although more far from the inoculation point, the positive detection also decreased. Well, at the end of the experiment, happened kind of the same. The frequency of detection get a little bit lower, and also in the stem section, when far from the inoculation point was the stem section. Also, the analysis showed that the main effect was due to Xylella fastidiosa strain, and in this case, also the stem section. And we have obtained also a higher colonization of the two strains belonging to subspecies pauca. So also a very important result in this experiment was that we don't have observed symptoms of the disease during the whole experiment. But this is very important from the second part of this research. Xylella fastidiosa infection was confirmed in all the plants we have inoculated. And with this, as we take advantage of the inoculated plants to develop an early detection system using proximal reflectance sensor. This type of sensor are based in the physiological indicators of the plants and their interaction with the light. Since when an infection occurs, it produces changes in the chemical and structural composition of the leaves before symptoms appearance. And this work was also tested in using an unmanned vehicle using also spectral information. I don't know what is happening. We have used two proximal sensors, the dual-X sensor that measure directly the chlorophyll, flavonoids, anthocyanin, and nitrogen level. And also we have used the SpectraPen sensor, which measures the spectral signature from 400 to 80 nanometers. And we have performed several measurements over time at 1, 3, 6, 9, 12, and 18 months after inoculation. And in each inoculation point, we have measured from 6 to 8 leaves at the inoculation point, and 5, 10, 15, and 20 centimeters from its inoculation point. So with the SpectraPen, we have obtained the spectral profile of each treatment, and we can calculate and estimate 70 pigment and physiological parameters that may be related with Xylella fastidiosa infection. And our analysis approach was using linear discriminant analysis that I'm going to show you in the next slide. Well, In this first slide, we observe the signature of the leaves of the spectral signature of each treatment. And here we can see anything, almost anything. But if we observe separated by each band, the blue band, green, red, and orange, we can see some differences between the treatments. Showing, and we can see several differences between the different inoculated plants and also respected to control non-inoculated plants. Well, in a second step, using now all the indices that we have calculated, this cluster analysis showed that first we observed a pattern by cultivar, but in each cultivar we can observe also some differences in between inoculated and non-inoculated plants. And when we perform the stepwise and linear analysis that we use to reduce the indices of vegetation used in this that can discriminate between the groups we have performed, in this confusion matrix, we can observe the global accuracy and the kappa coin that we have obtained in all the models performed. As we can see in all the models, we can the analysis selected 17 to 31 indices out of 74, and also we obtain a classification accuracy higher than 91.1% with a kappa coin higher than 0.86. So also the most important, apart from the good levels of classification accuracy at the good level of kappa coin, was in all the models were selected the same or similar indices or group of indices to discriminate between groups. And these indices were related mainly with pigments, carotenoids, chlorophyll, and flavonoids, the blue band, and others such as xanthophyll, chlorophyll fluorescence, and structural indices. Well, in this linear discriminant analysis plot, we can observe two main things. On one hand, we can observe the discrimination by cultivar in these canonical components. As we can see here, we can distinguish perfectly that each cultivar have different patterns. But also inside each cultivar, we can observe different patterns between inoculated and non-inoculated plants. And on the other hand, we observe a clear pattern between the objective of the research, which is to find different patterns between control and inoculated with Xylella fastidiosa plants. And also, the most important thing is that these canonical components are able to discriminate between Xylella fastidiosa infected and non-inoculated plants. The indices that more contribute to the models were carotenoids, flavonoids, the blue band indices, and also the xanthophyll. Also, another important result we have obtained between these two, between the non-inoculated and the inoculated plants, significant differences be at 1 and 3 months post-inoculation, and at that indices related with carotenoids, flavonoids, the blue band indices, and also the chlorophyll fluorescence. Well, to address the last, not the last, well, the third objective, that is, there are in real two objectives. We have performed on one side the modeling of the effects of temperature in vitro, and also the model of the dynamic oscillant vessels. Well, temperature is have a very important effect on bacterial development and also in the host plants. On one hand, previous research performed by Feld and Purcell on 6 Xylella fastidiosa strains from the United States showed that the optimal temperature of these strains was at 28 and 32 degrees, and while the survival was limited at 37 degrees and 5 degrees. Also, since Xylella fastidiosa is confined in the xylem vessels, it can be important to know how the xylem temperature differs from the external conditions. And that could be especially important in woody crops, because showed in other research, the plant tissues do not necessarily correspond directly to air temperature. So to address the first objective of this research, we in first place model the optimal temperature range for growth and biofilm formation across diverse Xylella fastidiosa strains in vitro, and we want to identify relationships among these Xylella fastidiosa strain-related variables with growth, planktonic, and biofilm formation to elucidate patterns across the response in temperature. And also, we want to evaluate, in this third objective, evaluate the lethal or the bacteriostatic effect of the suboptimal temperature on the selected Xylella fastidiosa strains. Well, for the first objective, we have studied 37 strains belonging to different subspecies, sequence times, geographical origins, and host plants. To try to avoid the highest diversity possible using Xylella. Well, and we have to do that, we have performed to address the first objective. We have incubated all these strains for 7 days in a range of temperature from 4 to 40 degrees. And to evaluate if the suboptimal temperature was the effect of this suboptimal temperature, we have taken the planktonic growth incubated at the range of temperature from 4 to 40 degrees and incubated them for an additional 7 days at the favorable temperature of 28 degrees. Well, in this first slide, I'm going to show you a general overview of all the results we have obtained. First, in first place, we obtained the growth curve that we can calculate the standardized area under the growth progress curve, and also total growth at the end of the experiment. Combine it to biofilm formation and planktonic growth. With the standardized area under the growth progress curve and the biofilm formation, we have performed the beta function models that consist in a model with the values of the temperature we have obtained across the range of temperature. These beta function models give us 3 parameters for each strain and parameter. And with all this information joined to the information, all the data we have performed, we have obtained in all the experiment, we perform a multivariate analysis using 46 variables. Well, in this first slide, I show you the beta function model obtained for growth. And we have obtained the general overview is that the optimal temperature growth range from almost 20 degrees to almost 30 degrees, but we see kind of pattern among subspecies. For example, the strains belonging from subspecies fastidiosa had a higher optimal temperature range ranged from 24 to almost 29 degrees, while Pauca has a lower optimal temperature range from 20 to 25 degrees. While the strains of the subspecies multiples had the widest range. For the beta function models for biofilm formation occurs almost the same for the strain studies of subspecies multiples, which have the widest optimal range temperature for biofilm, and also have a similar optimal range for the strains of subspecies Pauca. In contrast, the strain of subspecies fastidiosa had a lower optimum temperature range for bioinformation, indicating that a higher temperature promotes planktonic growth. Well, so as I say before, all these parameters estimated from the beta model joined with all the information in total we have 46 variables. We perform a factor analysis to aggregate these strains. And the loadings from the factor analysis are shown in this plot. And the first component are mainly, the contribution of the first component are mainly due to the total growth, the planktonic growth in all the range of temperature studied, and also the standardized area and the growth progress curve, let's say, at the optimal temperature. The second component, in contrast, the main contribution was due to the bioinformation at suboptimal temperature and also the optimal temperature for standardized area and the growth progress curve, while the third component, the major contribution was for the biofilm information at the optimal range of temperature. Well, and when we plot the scores of this canonical lot of this component, loaded component, we observe that a similar pattern that we have observed before in the beta function models. On one side of the plot, we observe the strains of Xylella fastidiosa, subspecies fastidiosa, and in the other, the strains belonging to subspecies pauca. And in the middle, there are all the strains of in the middle of these two, there are all the strains of subspecies multiples. Well, for the last sub-objective, to infer if the suboptimal temperature how affect to survival, we observed in the beta function models that a limited growth or no growth of bioinformation at temperatures lower than 10 degrees and higher than 36 degrees. So we want to know if this suboptimal temperature is bacteriostatic or bactericide for 23 strains. So we have transferred the planktonic growth incubated at the range of temperature from 4 to 40 degrees for another additional day at 28 degrees. And we have to evaluate that, we have calculated a recovery ratio of the total growth and bioinformation. And when the recovery ratio is higher than 1, we assume that these Xylella fastidiosa strains were able to recover growth or bioinformation. In contrast, when ratio recovery values are lower than 1, it means that the temperature exposure had a lethal effect. And the main results we have obtained for growth were that those strains incubated in the previous experiment at the temperatures lower than 10 degrees do not limit bacterial viability. In contrast, those strains incubated for 7 days at temperatures higher than 36 degrees had a lethal effect on growth. Also, for bioinformation, obtained kind of similar results. In these images, we can observe that in the range from 4 to 10 degrees and 36 to 40 degrees, we don't obtain any bioinformation. Although when incubated those strains at the favorable temperature of 28 degrees for another 7 days, we observed that again the strains incubated at temperature lower than 10 degrees were able to recover that bioinformation. While in contrast, we don't observe the bioinformation in the strains incubated at temperature higher than 36 degrees. But also we want, in a last experiment, we want to test what happened when we incubate the strains, some of these strains at lower temperature times, at lower times, sorry. And what we have observed at 6, 12, 20, 24, and 72, and 120 hours after the inoculation was that the main results was obtained, not the main results, the main decreases of the growth was obtained between 24 and 24 hours after inoculation and 72 hours after inoculation. And also we have used a qPCR viability test called the PMAX. And in this example of the dono and temecula, we can observe how the cell viability was maintained at temperatures lower than 10 degrees, while the cell viability decreased at 36 and 40 degrees between 24 and 74 hours after the inoculation, obtaining less than 11% of viable cells at these temperatures. Well, for the last objective, as I explained before, we model the temperature of the xylem vessels in the Mediterranean area. For that, the objective was to describe the temperature dynamics within xylem vessels and the soil rot interface in olive and almond tree in the Mediterranean climate, and determine the relation with the air temperature. And also, we want to characterize the influence of the type of plant tissue, the depth of the xylem tissue, or the tree orientation in the temperature variations. For that, we have selected several trees in an almond ocar and an olive tree ocar located in Córdoba in Spain. In collaboration with the Department of Agronomy of this research center, we have installed several thermocouples to measure the temperature in range, at 2 depth in trunk, and also at 20 centimeters depth in the soil rocks interface, and also in 2 orientations. And the main results we have obtained for minimum temperatures was that the temperatures was the models show that the air and xylem temperature was linearly related, with increase of a slope around 1, which means that the temperature of the air and the temperature of trunk and branches was very similar. In contrast, when we observe the results for maximum temperature, which occurs from April to September, we observe that the xylem branches have the same behavior, was very similar to air. However, the xylem trunk temperature was lower than the air, and the obtaining from 4 to 8 degrees lower than the air. And we obtain the same in the models, obtaining the lowest slope in summer at 1 centimeter depth. And when we observe the results for soil, we observe knowledge oscillations for in winter or neither in summer. And also we observe that for minimum temperature, we observe a high increase of around 7 degrees respect to the air when the minimum temperature occurs, and around decreases of around 10 degrees when maximum temperature occurs. And this relation between the temperature of the real soil sensor and air was lower in winter and summer, as we have observed in the observation of temperatures. And this information could be very important since in the Mediterranean area, we don't have observed that the temperature in the trunk, in the xylem trunk, or neither in the interface, in the soil rot interface, where Xylella is able to move in winter, to overwinter there, could limit the survival of Xylella fastidiosa in our conditions. Finally, to conclude, the take-home message are that the phenotypic traits and genomic features, including plasmid of each Xylella fastidiosa strains, may confer virulence factor associated to pathogenicity and adaptation to new environments. Also, the pathogenicity experiment showed that the differences in colonization patterns among the Xylella fastidiosa strains studied from different genetics and geographic origins observed in the studied pathosystem highlight the risk for new disease outbreaks. Also, the spectral data measured on leaf enable the early detection of Xylella fastidiosa infection in asymptomatic plants, providing a valuable tool for disease monitoring. And finally, the last objective showed that the optimal temperature was determined between almost 20 and almost 30 degrees for growth and 15 and 30 degrees for bioinformation. And we also have observed subspecies specific thermal responses. The incubation temperature of 4, 6, and 10 degrees do not impair the recovered growth and bioinformation, while at 36 and 40 degrees affect to cell survival. And finally, the buffering effect on temperature within the trunk, healing tissue, and growth create a more conducive environment for Xylella fastidiosa survival. And well, yeah, thank you very much for your attention, and thank you very much for all my research group, and especially to Juan Antonio and Blanca, because without them, this all work can be almost impossible. And thank you very much.
Thank you very much, Miguel. Excellent presentation. Thank you. A lot of data to show. So thank you very much. And now we open the floor to questions. Again, you can write it in the chat, or you can raise your hand and speak.